describing how aldosterone and ANP, atrial natriuretic peptide, regulate plasma sodium levels, include an explanation of the stimuli that release these hormones. Also include an explanation of the stimuli that release these hormones. Okay, that was written twice. So the first thing we'll look at is sodium balance, and then we'll get into potassium balance. They are combined, so I kind of mentioned them together. And we'll see what that means here in a little bit. So we'll start off with some terminology as it applies to sodium plasma levels. So when you think about high sodium plasma levels, that's termed as hypernatremia. When you think about low plasma sodium levels, that is termed as hyponatremia. And so sodium is really, really important, as you would have imagined, just having gone through several units thus far. We talked about the sodium potassium pump. It's on the basolateral surface of most of our cells, really important in osmotic pressure. So it's going to help create that gradient that will then allow water to flow according to osmosis. So sodium is the primary solute in our ECF, and that is heavily contributed to because of the sodium potassium pump that's pumping that sodium out of the cells. It's also important for excitable cells. So things like muscles, neurons, they are reliant on sodium for the generation of that action potential that we talked about, right? The main ion that generates the upstroke of an action potential is sodium. So sodium is really important, but it's also important to be very heavily, very tightly regulated. Even a slight change in sodium plasma levels can be detrimental. Hypernatremia can cause things like seizures, it can cause coma, it can become fatal. Hyponatremia can cause fatigue, lethargy, other things on the other end of that spectrum. So it's really important to have sodium in a tight balance, and we'll talk about how that is brought about with the help of the kidneys. Okay, now as far as the mechanisms by which sodium is regulated throughout the kidney tubules, sodium is freely filtered across the glomerulus, so there's no obstruction there. And then it is absorbed across the proximal tubule, distal tubule, and collecting duct. About 70% of that sodium is reabsorbed. And so there is some sodium left in the filtrate that becomes urine, right? The urine is slightly salty, if you will. And so that can be regulated by the help of two hormones that we're going to speak about. So it's not secreted at all. We'll see it's different for potassium. Potassium is secreted, but sodium is not secreted at all. It's only reabsorbed. And that reabsorption is regulated by aldosterone. Aldosterone is going to increase sodium reabsorption, so bringing more sodium back into the vasculature. It's also regulated by ANP, so atrial natriuretic peptide, and that's going to have the opposite effect of sodium. So that's going to cause sodium to be not reabsorbed, and that is going to allow for more sodium to be lost in the urine or excreted as a part of that filtrate. Most of the regulation happens at the principal cells. This is in the distal tubule and the collecting duct. Here we mentioned that there's regulated reabsorption, and so this is how that reabsorption is regulated under the influence of these two hormones, okay? There's active reabsorption. The mechanisms we'll get into in just a bit, but the mechanisms rely on that sodium potassium pump on the basolateral membrane. So because we're pumping sodium out, we create the gradient for sodium to come into the cell passively. So it's really important to have those active pumps on the basolateral membrane in order to facilitate that sodium reabsorption from the filtrate and back over into our peritubular capillaries, okay? All right, so here is what that mechanism looks like. This is again a review at this point. We talked about the mechanism of the proximal tubule. We've got sodium coming in by secondary active transport, and then we've got it leaving the cell by the sodium potassium pump. And again, just to really reiterate this, it's really important for you to know these mechanisms. So these will be tested. You should be able to explain how does sodium get into the apical surface of a proximal tubule cell, right? That's secondary active transport. How does sodium get across the basolateral surface of a proximal tubule cell? That is the active pumping with the sodium potassium pump. The distal tubule is slightly different. So it's coming in via secondary active transport, but the way that we can up and down regulate this, the way that aldosterone can play a role in increasing reabsorption versus decreasing reabsorption is through these leak channels for sodium. So there are some leak channels for sodium that are on the membrane of the distal tubule and collecting duct only. And so aldosterone is going to insert more of these leak channels. It's also going to insert more potassium channels. We'll talk about potassium later on. But aldosterone inserts more of these leak channels into the apical surface of the membrane. And so we can reabsorb much more sodium than we would with just secondary active transport alone, okay? And then across the basolateral surface, we still have that sodium-potassium pump. So exclusive to the distal tubule and collecting duct, we've got the upregulation of these sodium channels via the influence of aldosterone, which helps to increase that amount of sodium reabsorption. So let's talk a little bit about aldosterone. It increases sodium reabsorption. It is a steroid hormone. So it's secreted from the adrenal cortex. So if you remember from the endocrine system, we talked about the outer part of the adrenal gland secreting mineralocorticoids, glucocorticoids, androgens. A part of one of those mineralocorticoids is aldosterone, right? That's one of the specific examples. And so it's going to act on the principal cells of the distal tubule and collecting duct. What it does is it increases the number of sodium potassium pumps on the back, on the basolateral surface here. It also increases the number of open sodium and potassium channels on the apical surface. So we'll talk about how aldosterone influences sodium in our second objective here, but essentially it's going to input more leak channels, both sodium and potassium on the apical surface. It's also going to input more sodium-potassium pumps on the basolateral surface. So in doing that, it's going to facilitate more reabsorption of sodium, and as we'll see, more secretion of potassium. We'll talk about that again later on. Now, aldosterone is under the influence of the RAS system, so the Renin, Angiotensin, Aldosterone system. So this is kind of this cascade that's created with these three compounds. We've got Renin, which is the first stimulus here coming from the kidneys. We've got angiotensin, which is what is converted from Angiotensinogen, and then that eventually becomes aldosterone. So aldosterone is kind of the end product of some other things that are happening upstream. So the first thing that happens is the granular cells of the juxtaglomerular apparatus. So we talked about these cells initially, the cells that are at the afferent arteriole. So they're part of that JG apparatus that we talked about, or the juxtaglomerular complex, the complex of those three cells. Specifically, the juxtaglomerular, also called the granular cells, are the ones that secrete renin. So they are going to secrete renin. We can kind of see them here in this, this little schematic here. Now, always circulating in our plasma, in our blood, is Angiotensinogen. And by the suffix there on that word, we can tell that this is the inactivated form of this compound. So we've always got this Angiotensinogen in our blood in its inactivated form. This is being secreted by the liver. And under the influence of renin, that Angiotensinogen can be converted to angiotensin 1. Okay, so that's kind of a precursor to the next thing that we'll talk about. So we've got this angiotensin 1 circulating in our plasma. Now, under the influence of another enzyme, this is ACE, so angiotensin converting enzyme. This is being secreted from the capillary endothelial cells. That angiotensin 1 is now converted to angiotensin 2, and then angiotensin 2 is going to directly stimulate the adrenal cortex to secrete aldosterone. So this is kind of a cascade of events that's happening under the influence of renin from the JG cells of the juxtaglomerular complex, as well as the influence of ACE, angiotensin converting enzyme, from the capillary endothelial cells. Okay. So let's kind of describe that here one more time. So we've got the liver always secreting angiotensinogen in its inactivated form. So this is always circulating in our plasma, very similar to how those clotting factors in their inactivated form are always existing in our plasma. Now, what's going to kind of kickstart this whole thing is renin that is coming from the juxtaglomerular cells. And I'll talk about some of the stimuli for this renin secretion coming up. So the first thing that happens, the granular cells here, the JG cells of this complex will secrete that renin, and that will initially convert angiotensinogen into angiotensin 1. The next thing that happens is the conversion of angiotensin 1 to angiotensin 2 under the influence of ACE. And ACE is secreted from our capillary endothelial cells. This is the angiotensin converting enzyme, and this can catalyze that conversion of angiotensin 1 to angiotensin 2. And then angiotensin 2 is going to directly stimulate the adrenal cortex. So again, think about the adrenal gland, think about that outer lighter region, the cortex, that region being stimulated to secrete our aldosterone under the influence of that angiotensin 2. All right. Okay. Now let's talk about some of the other things that angiotensin will do. So we just talked about one of the main things that it does, which is to stimulate that adrenal cortex to release aldosterone, but it also does some other things that are going to help increase blood pressure as well. So the first thing it does is it's going to cause vasoconstriction, and we talked about this effect when we looked at the arterioles back in the vascular system in chapter 15 or 16, I believe. So we did speak about angiotensin 2 that will encourage vasoconstriction, and that will help to increase mean arterial pressure. The second thing here we just encountered, right? It's going to stimulate the adrenal cortex to secrete this aldosterone. That's going to insert more of these leak channels into the apical membrane of the principal cells of the distal tubule and the collecting duct. That is going to reabsorb more sodium. That will help to increase the extracellular fluid osmolarity. In other words, the saltiness, the sodium concentration of that extracellular fluid. And so that'll be the second thing that angiotensin 2 does. The third thing angiotensin 2 does is to stimulate the posterior pituitary to secrete ADH. And this is being done directly by angiotensin 2, but it is also being done indirectly because when we reclaim more sodium, we increase our sodium osmolarity. And if you remember us speaking about those osmoreceptors, that was another stimulus for ADH secretion. So ADH secretion will increase both directly because of angiotensin 2, but also indirectly because we're increasing plasma osmolarity. And that is another trigger or stimulus for ADH as well. Okay, and then that ADH we talked about, its effect is going to go to those same principal cells, insert more of those aquaporin 2 channels, allow for more water reabsorption in the distal tubule and the collecting duct, and that will help to increase plasma volume. That is what is going to correct our mean arterial pressure or our blood pressure. Okay, so we kind of take a sideways route here, and I talked about this relationship before. The way that the body moves water is to move sodium first. So a part of the back route of doing this is reabsorbing more sodium, increase the plasma osmolarity, and then having a higher plasma osmolarity allows for more water reabsorption through this ADH connection. And then that can correct our plasma volume or increase blood pressure. And the last signal here that is sent by angiotensin 2 is to the thirst centers in the hypothalamus. That is going to increase our thirst sensation, right? Requiring more water. And so we're going to drink more water if we're more thirsty. That will directly increase plasma volume, which will also correct mean arterial pressure. Okay, so these are the 4 things that angiotensin 2 does. And then one of those main things we just described was really involved in that RAS system. So it helps to convert renin coming from the kidneys to aldosterone through that angiotensin 1, angiotensin 2 connection. All right, let's talk about the stimuli for renin. So what makes the kidneys decide to secrete this renin that will then convert that angiotensinogen into some of the other things that we just talked about? So the stimuli for renin are threefold. One is decreased pressure in the afferent arteriole, and that kind of makes sense. If we have a drop in pressure in the afferent arteriole, this is going to cause a drop in GFR. We're going to do things to start reclaiming more sodium, reclaiming more fluid, to increase mean arterial pressure, to increase GFR, to increase the pressure in our afferent arteriole, right? So the first stimuli or stimulus is going to be a drop in pressure in our afferent arteriole. The second trigger is going to be the sympathetic activity. So this is detected by the baroreceptors if they have a lesser, or if they have less stretch being detected from those baroreceptors that are checking for our mean arterial pressure. That will be another trigger for sympathetic activity, and that will be another trigger for renin release, again, in an attempt to correct that drop in mean arterial pressure. The third stimulus for renin is if there is a decreased amount of sodium and chloride coming through in the distal tubular filtrate. So if the cells of the distal tubule, and although it's not specified here, the macula densa cells are going to sense this, right? Remember, the macula densa cells are the cells in the distal tubule. They are going to sense that there's not a lot of sodium in the distal tubule and there's not a lot of chloride. And so what they do is send signals to the other cells, remember those paracrine signals, to these JG cells for them to secrete renin, and then that renin will help to correct that imbalance. And because renin is going to lead to aldosterone secretion, because of that RAS connection, renin converted to angiotensin 1 and then 2 and then aldosterone, that aldosterone will allow for more of that sodium reabsorption. Okay, so this is a more clear illustration of those three stimuli for renin. So the three things that will cause renin release is a drop in afferent arterial pressure, right? A drop in the baroreceptor stretch, which will stimulate sympathetic nerve activity, and then a drop in GFR, which will lead to less sodium, less chloride coming through the distal tubule. The macula densa cells will pick that up and send signals to the JG cells. They will secrete renin, and because of the other things that we spoke about, renin will eventually lead to angiotensin 2. Angiotensin 2 will bring about those four mechanisms that we described. So these four mechanisms, which will help to correct that mean arterial pressure. And then in a more direct way, aldosterone will also come into play, which will help to correct that sodium.